So next video is, this will be our video clip number 5. So it's about parametric or non-parametric statistical tests. Okay, so what are parametric tests? So these are based on assumption about the shape of distribution and form of distribution of features. So it's usually applied to those data who have normal distribution or who follow the Gaussian curve. Okay, an example of simple parametric test is student t-test. And what are non-parametric tests? So these tests do not rely on assumptions about the distribution. So all those data which don't follow the normal distribution or Gaussian curve, they're called non-parametric tests. There's less scope for erroneous of improper use of non-parametric test compared to parametric equivalent, or that example is Mann-Whitney U-test. Mann-Whitney U-test. And why not always just use a non-parametric test if it makes no assumptions about the underlying data? So non-parametric tests have two drawbacks compared to their parametric equivalents. First, they are less statistically powerful, which means there is a smaller probability that the test will tell us the two variables are related when they are in fact related to each other. And consequently, non-parametric tests require a slightly larger sample size. So in sample, so first they're statistically less power, they need to be bigger sample, which is always difficult to gather a bigger sample. Other drawback is that the results of non-parametric tests are less easy to interpret, they're difficult to interpret. So they also tend to rely on ranking the values in data set rather than using the actual data. So what test should be used for categorical and non-normal and normal data? There's a small table on page 12. So for categorical data, non-normal are compare median and normal compare mean. So for categorical data or parametric data, we will use, if we are studying one sample, chi-square, two groups unpaired, again chi-square, two groups paired, then we have McNemar's test, and more than two groups unpaired, and then we use chi-square, more than two groups paired, so McNemar's. So in other words, categorical data has two tests, chi-squares and McNemar's test. Okay, let's go to the non-normal compare median. So if you remember the median, center of the data. So if we have one sample, the test will be Wilcoxon single rank test. If we have two groups but unpaired, then we will use Mann-Whitney test. If we have two groups and they are paired, then we will use Wilcoxon matched pairs test. And when we have more than two groups unpaired, we will use Kruskal-Wallis test and ANOVA test. Okay, so remember Mann-Whitney, Wilcoxon, ANOVA. And if you have more than two groups which are paired, then we use Fredman's test. And then the normal compare normal data with compared to mean. So if we have one sample, we will use one sample t-test. If you have two groups unpaired, t-test, two groups paired, t-test, more than two groups paired, ANOVA. Okay, so this was that, or the test. Then we go to the power. What do you understand by the term power in medical studies? So it is the measure of the ability of the study findings to conclude that an observed effect really occurred rather than simply being the result of inaccurate observations or experimental error. Results are not due to inaccurate observations or experiment, it is the real effect. It is related to the sample size. Generally, the larger the sample size is greater the study's power. And large studies may detect small differences that reach statistical significance but are of dubious clinical significance. Explain the difference between clinical and statistical significance. Statistical significance indicates that the study findings are not likely to have occurred by chance alone. So there is some factor behind that. This differs from the clinical significance, which is the clinical importance or meaning of the studies. Large studies may find tiny differences between the patient groups that reach statistical significance but are unlikely to have a great clinical significance. If something is statistically significant, it's not always a rule of thumb that it will also have a clinical significance. Conversely, small studies may identify larger clinical differences between the patient group that fail to reach the statistical significance but would be of great clinical importance if true, warranting further. How do we undertake a power calculation for a randomized controlled trial? So this should involve assistance from an experienced clinical statistician and realistic data regarding the proportion of patient with the outcome of the interest in the control will be required as a baseline. So we have to decide before that how much will be the power of our study. And assumption that has to be made about the expected effect size of the intervention under investigation. Larger the effect size estimate, is smaller the number of patients required in each arm of the trial. So effect size. So larger the effect size, smaller the number of samples. Or the large effect size may be unrealistic. And this in turn may result in the incorrect conclusion that intervention has no effect. Numbers needed in each arm of this trial are determined using the pre-calculated tables for common statistical tests. Okay. So what do you understand by the term stratification? So stratification is a method of ensuring an equal distribution of key confounding factors between the arms of a randomized trial. It avoids relying on the chance to produce an even distribution of these key confounders. Stratification is particularly useful in small trials which are particularly vulnerable to an uneven distribution of key confounders occurring by chance. So small trials we can use stratify. And then is let's finish here.